is a really great alternative to PyMol and ChimeraX for investigating molecular structures, such as the structures of proteins and nucleic acids. A few things make it great. One is that it's way easier to use. Two is that it's totally free. ChimeraX is free, but PyMol, you have to have like a registration and things like this. This is totally free. It's web-based. You can access it anywhere without even having to download any software. What's really, really cool about it is that although it can't do quite as much as ChimeraX and PyMol in terms of things like making publication quality figures and doing really complex functions, it can do things that those can't. One is that it's integrated with databases, so it'll automatically, you just put in the PDB session code and it'll automatically bring in all sorts of information, things like post-translational modifications, conserved domains, really, really cool stuff. It also allows you to basically save HTML code that you can then access the session again and send it to someone else so that they can view what you're viewing. And you can save, basically embed code into another web page if you want to have an interactive image, an interactive structure that people can play with on the web page. When you open up iCn3D, you're going to get a screen that looks something like this. And the first thing you need to do is tell it what you want to look at. The common way you're going to do this is by giving it the PDB accession code, in this case, 1HEW. This is the crystal structure for hen egg white lysozyme, so this enzyme that breaks down the sugars in bacterial cell walls, and that we use a lot in the lab to do that to help us purify proteins out of bacteria, say. And in this case, it's bound to an inhibitor that's going to mimic that sugar. What we're going to do is we're going to put in this PDB code. You can also put like an AlphaFold uniprot structure code in there to have like a structure that was predicted by AlphaFold. What we're going to do is we're going to go ahead and load the biological unit. It gives you a little bit more information about this, but basically the biological unit is going to be the biochemically active form of a biomolecule. So if you load the asymmetric unit, that might have something like 2 copies of the protein, but it's not actually working as a dimer. It's just like a crystallographic artifact type of thing. Whereas the biological unit, this is going to be like the way that it's the biochemically active form of it. So we're going to go ahead and we're going to load that biological unit. Okay, so we're going to by default see something that looks like this. And I'm going to show you a bunch of different ways that we can represent this, as well as how we can kind of like show connections between this inhibitor molecule and the protein. First, you might be wondering what those yellows are. We'll get to that in a second, but those are going to be kind of like disulfide crosslinks between different regions of the protein. It's one of the reasons why lysozyme is so sturdy and good for a bunch of different things is because it has those disulfide crosslinks. The first thing I'm going to do is I'm actually going to change the background color. If I go to Style, Background, I'm going to make this transparent. And I also want to be able to see the sequence. If I go over to Analysis and then Sequence and Annotation, well, now I'll have this side box pop up. Now, you can see the sequence in pretty much any tool that you use to look at structures, be it PyMol, be it ChimeraX. But one of the really cool things about iCn3D is that you're not just getting the sequence, you're also getting all of this other functional and annotation data. Basically, although I gave it a PDB code, it didn't just fetch me the PDB file, it fetched me something that's like an MMDB file, which basically has a bunch of other annotation database information attached to it. iCn3D is like made and hosted by the NIH and the NCBI. So it has access to all of those different databases. And so it automatically pulls them all in. And I can show all of those different things, like if I wanted to show functional sites, if I wanted to show post-translational modifications or disulfide bonds, say. Those are what those yellow things are. See, if I click on this, now it's going to highlight these cysteines that are making those disulfide crosslinks. And so when you when you select something, it's going to be highlighted in yellow. By default, if you wanted, you can change that highlight color, that highlight style, or you could toggle it on and off. So right now it's toggled off, even though I'm still thinking about that selection. Okay, so I want to clear that selection though, because I'm not going to be worried about that right now. We'll talk more about how you can use these selections in order to represent parts of the molecule in different ways than others. So right now, we actually haven't seen the sequence, but we could do that if we go to details. Note that because I have disulfide bonds still listed, it's going to show that as a separate track. It also has domains listed, so it's going to show that as a track. Now, in this case, our domain is basically the whole thing of the protein, because this is basically just the conserved lysozyme domain with a little tiny bit at the ends that's extra. And so what we're going to want to do is we're going to go ahead and we're going to toggle off that domains. And for now, we're just going to toggle off the disulfide bonds. And now all we're going to see is this protein chain. Now in this structure, we just have this single chain, and it's a pretty small one, which makes it nice for looking at, just 129 amino acids. And it's going to have this, because we have a single one, we're only going to have a chain A. If you had multiple chains, then you would have like B, C, D, E, etc. And you can select it by just clicking on it. Now we also have some other things. This one is going to actually be our water. So right now, by default, it's not showing the water. But if you go over here and you can go to style water, I can show it as spheres, I can show it as dots, or I can hide it. For now, I'm just going to hide these waters. And note that the amount of water you have in the structure is going to depend on kind of like the resolution of the structure and whether the people like modeled in water molecules. We're also going to have this. So this is going to be our inhibitor. It's like this tri-NAG thing. So basically it shows like count 3 NAG, 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 but really it's this one connected molecule that you can see here. What we're going to do later is we're going to show how we can show interactions between this inhibitor and the protein. But for now, let's just look at how we can show the protein in different ways. I'm going to go ahead and I'm going to go back and I'm going to select that protein. And you can see up here that I'm working on the selection. If I toggle this off, now I'm affecting everything, even though the protein is still selected. But what now I just want to do, be working on the selection. What I can do too is if you want to see more options in a toolbar setting, you can have them pop up under here, or you could go to these different tabs for all of the different options. The first thing we can play around with is how we want to actually represent this structure. So right now it's in ribbon diagram. And when you're in ribbon diagram, you have this thing where basically the alpha helices are going to show up as these kind of these ribbon looking things. And then you have the beta strands are going to be these arrows. And then you have in between the things that are less, don't have that defined secondary structure are going to be kind of these loops and coils and stuff. Those are just going to be shown as these thick lines. What we could do is if we want to really emphasize those features, we'll see later how we can actually color it by those secondary structure elements. But first, let's look at how we can display this structure in a couple different ways. One thing to note is that you can do things like show and hide the chains, side chains. So right now the side chains are hidden, but if I wanted to, I could do things like show them in stick. But now I hope you can see why we often keep them hidden, because it can get kind of hard to see what's what when you have them shown. So what we're going to do is we're just going to hide the side chains. And then later what we'll do is we're going to go ahead and we'll introduce some of them, just the ones that are going to be interacting. But right now, let's just leave it like this. You can also show it in different ways. If I wanted to, I could do things like ball and stick. I could do things like sphere. I could hide the whole thing in all together, but right now let's just go ahead and we'll add it back to the ribbon. But what I do want to do is I do want to change the coloring. So right now by default, it's kind of just this magenta. If I want to change it to a different single color, I could do that here. Or what I could do is I could color it by various features. A common feature like rainbow is basically going to go and it's going to color it from the N terminus in this like purpley blue to the C terminus in a red orange. So you can see that the protein is kind of going like this all around. I still think it'd be really fun to have like an amusement park where you had like a roller coaster that is shaped like a protein. So if anybody's interested out there, that'd be really, really awesome. But you can also see that we have these disulfide crosslinks that are actually linking together regions near the N terminus and near the C terminus, which is pretty cool. Okay, but anyway, that's one way you can represent things. And that'll tell us information about kind of how the protein is laid out as it's getting made, but it's not going to tell us much about what are the amino acids within those different regions. So in order to get more information about that, we have a couple different options. If we want to, we could actually just like color it by residue. And so now it's just going to color each of these a different color depending on what amino acid it is. And again, right now, we're just showing the backbone. We're not actually showing the side chains, but it would make more sense to look at it this way. If you had the side chain showing, you could actually see, oh yeah, that really is a leucine or a valine or whatever. And so if we wanted to, we could go ahead and we can do side chains and we could show those side chains. Let's do a stick. And now you can see that, yeah, the ones that are, the arginines really are that dark blue and that sort of thing. Okay, but first, let's go ahead and we're going to change and we're going to hide the side chains again. And instead, let's show it in a different coloring scheme. Let's go ahead and we'll show it by secondary. So here we can actually do things like we're highlighting those strands and those coils and those sheets and those helices in different colors. And so that's another way that we can color it, and another way we could highlight those features. And again, you could do this even if you weren't showing it in ribbon mode, and then it would make it easier to see those features really are there. Okay, another thing you could do is you could color it by charge. So you can see here that by convention, a positive charge is going to be blue. So if you have like a lysine or an arginine or a histidine, those are going to show up as these blue. And then if you have something like a glutamate or an aspartate, something that is negatively charged, these are going to show up as red. And then you have your neutral things that are going to be this gray color. You can also color it by hydrophobicity. So this is a cool one. So like, how much does it like to hang out with water? If water likes to hang out with it, it's going to be hydrophilic and it's going to be colored white. And you can see that the white regions are kind of like on the outside of the protein. This really great example is in this alpha helix. You can see that the white areas are over here. And then on the inside, you have these more green areas, and the green areas are going to be hydrophobic. And so they're kind of hidden away from the water in the center of the protein. We can also color it by things like B factor, which is a kind of measure of how confident the data is in a region. If an amino acid, say, is flopping around a lot, it's going to have a higher B factor. And so it is going to be the resolution in that area, basically the ability to really accurately model it is going to be more iffy in that area. So this structure actually it's an older structure, so it doesn't have the electron density map associated with it. But if it did, what you'd be able to do would be go down to here at electron density, and you could display the map here. These days, people have to have their maps deposited, but this structure, I think, is from like 1992, so they don't have this structure here, or else I would show it to show you what I mean. But you can also color it by other things like solvent accessibility, structure domain. You can do it by domain. If we had multiple domains in here, we could do it like that, and various things. For now, let's go ahead and we're just going to go and back and color it. We're just going to color it back in that by secondary structure. And now what I want to do is I actually want to model the interactions between our inhibitor and our protein. First, I want to show you a little bit of the surface so you can actually see what we're dealing with. So instead of showing the inhibitor right now, it's in this like stick mode. What I want to go ahead and do is I'm going to display it in spheres. So if I go to style, before I was dealing with the proteins, but right now I'm going to be dealing with the chemicals. So I go to chemicals, and what I want to do is I'm going to show it as sphere. So now you can see, wow, this thing is actually a lot bigger than I was thinking of before. Or what I could do is I could go to style and I can apply a surface, or right now I want to actually go and I'm going to select my protein and not that. And maybe I want to go to style, surface type, and I want to show the solvent accessible surface. So now you can see, yeah, there's that pocket that the drug is binding into. And we'll look at how we can do this for just a region of the protein. But for now, I'm going to go and I'm going to remove that surface. And note that we could also do things like the molecular surface, which is going to, maybe I need to remove that surface. You can also do things like display like the van der Waals surface or the molecular surface, which is basically going to be defined more by the atomic radii. But you can see that you've got that binding pocket right in there. The reason why we saw it more fuzzy with the solvent accessible is because you can see that there are actually still water channels throughout the protein, even though we're displaying it in this way. So proteins really do have a lot of like room for water flowing through them. Lysozyme is actually a pretty compact one, so there's not that many water channels. But for other molecules, you see a lot more solvent accessibility inside of the protein itself. You can also do things like change the surface opacity so you can see some of the sticks underneath, the lines underneath. But right now, we're just going to go ahead and we're going to remove the surface. And what we're going to do is we're going to go back and we're going to style our chemicals. We're just going to do this as stick mode again. But oops, I wasn't selecting on it. So I need to go and I need to select that. Now I go to style chemicals and I can do this stick. And note that you might get kind of confused because you go to the menu and you can see that, okay, well, I had it before, I had it selected a stick, but it wasn't shown a stick. The check mark is basically what you click last, even if you weren't actually on the thing that you were trying to change at that point. So you need to click it again, even if it's already clicked, if it's not showing up. Now what I want to do is I want to go ahead and find the interactions and show them. So I'm going to take, right now I'm selected on this inhibitor, this NAG. And what I'm going to do is I'm going to go to analysis and I'm going to go to interactions. It's going to ask me, what do I want to check for interactions between? And it'll also choose me the types of interactions that I want to look for, as well as the thresholds. So you can see that because salt bridges or like ionic bonds can act over a longer distance, it's going to have like 6 angstrom cutoff. But for something like a hydrogen bond, which is this, which needs to be closer, things need to be closer together, it's set to 3.8 angstrom. And if I want to be even stricter, I could set things lower for various interactions. I need to tell it what I want to find interactions between. So I'm going to go and I'm going to select this NAG, and then I'm going to select this HEWA, that one HEWA, which is the chain for our protein. Now what I'm going to do is I'm going to display the 3D interactions. And up on here is going to pop up. It's going to have found the interactions between the protein and this molecule, at least the predicted interactions, and it's going to display them as dotted lines. Let's take a look at one of these. For example, we can see here we have an interaction between this inhibitor and what looks like an asparagine 59, but we can't really tell where it's interacting because we're looking at just the backbones and we don't have the side chains displayed, except in this one case, which I'm not sure why is actually displayed now. But anyway, we want to display them all. And so what we're going to do is we're going to chain, right now we have them deselected. And what we could do is we could go over here and we can go to style, proteins, sidechain, and we want to show these as stick. And so now we can see all of those different amino acids that we're interacting. What we could do too is if we go to this like 2D interaction network, basically what it's going to do is it's going to show us this sort of map type of thing where we can see the predicted types of interactions between the different residues of the protein and the structure of the inhibitor. What I also want to do is to kind of highlight more because it's not just making a bond to the residue, it's making a bond to a specific part of the residue or interaction with a specific part of the residue. Like this one, for example, what is it actually interacting? What you could do is it makes it easier to see things. If you go to now color, we're going to color it by the atom. And so now this is going to be colored as well by the way that's the same way as the inhibitor, in that we have the carbons as gray, the nitrogens as blue, and the oxygens as red. And what you can see now is, oh yeah, we've got a really nice looking hydrogen bond between this oxygen and this asparagine this asparagine nitrogen of the backbone. And so you can see that some of these interactions are through the backbone and some of them are through the side chain. But all of these interactions together contribute to help keep that inhibitor bound. And you can do all sorts of different fancy things to show it in different ways. You can go ahead and you can measure the distances between these. You can do things like if you wanted to display the surface, say we want to display style, surface type, let's go ahead and we're just going to display the surface for just those regions. And now because they're colored the same way, it's kind of hard to see. But we would be able to, there are different things that we could do in order to show kind of like the binding pocket. In addition to just showing the interacting residues, we could have done something like maybe what we want to do is we want to go, So you can use if you're using a mouse pad, you can use 2 fingers, press down with 2 fingers if you want to move things like side to side. 1 finger with your thumb to kind of like scroll around and pinch and to zoom in and out. And there are also commands you could do with like a mouse. If you want to focus on a selection, there are ways that you could do this. So view, you can kind of like zoom in on a selection if you want, or various other options. You can also do things like view, you can do a slab for a selection, where basically it'll let you just look at layers. If you pinch in and out, or various different things that you could do. So you kind of just have to play around. Okay, but one of the things I wanted to show you was that what I could do is right now I'm just going to clear my selection. And I don't want to look at these interactions anymore, but I do want to look at the sequence. And what I want to do is I'm going to select, I'm going to change that selection. And now I'm just going to be selecting. And now I'm just going to be selecting this inhibitor again. And instead of just showing these interactions, what I want to do is I'm going to go and I'm going to file, I'm going to select by distance. And so now I'm going to select this, I want to select the chemical. See, now I have these different ways I can select all of those different things that I've computed. But right now I just want to go and I want to select this inhibitor again. And then I want it to show me the things near it this way. And I'm going to set a radius of 6 angstroms. Now what I'm going to do is I'm going to have it select those things. And so you can see now it's going to be selecting more than we had selected before. And now what I could do is I could go and I could style it, and I could do something like add a surface on there. And now you can see that I can see that binding pocket for the drug. Well, if I want to go and I can select this like E35, which is going to be like a catalytic residue. If I select that, maybe I want to, well, first of all, see where the heck it is. So if I go to view and I can do to center selection. And now, because I have it on slab still, if I take that slab off, should be able to see this a little easier, or maybe not. Where is that selection? View, I'm going to center selection again. Okay, and now what I'm going to do is I'm actually going to style. I'm going to remove that surface so we can see things a little better. And here's what we're looking at. And because it's not shown the side chains, that's why we're having a hard time seeing it. If I go and I go to style and I go to side chains and I can show the side chain for this amino acid, okay, now we can see it better. And maybe what I want to do so I don't lose it again is I'm going to go and I'm going to go to analysis and I'm going to label it. For now, I'm just going to label it by residue. And right now it's labeling it right here, but there are different ways that you could label it as well. And I can also change the label scale if I wanted to label it really, really big, say, and et cetera, et cetera, et cetera. And I could label the individual atoms instead of the residue if I wanted. So all sorts of really, really cool ways that you can represent things. So you just really have to play around to figure out what you want to display in different ways. You can also just one other quick thing is if I wanted to go and look at all those selections, well, I can select it by a bunch of different things, but I can also make custom selections. You can use set operations. So you can say like, I want it in this set, things that are in this set, but not this set, or things that are in this set and this set. So there's a bunch of different ways that you can do things. What you'll also see is that at the bottom, what it has is, it has actually, it's been everything I've been clicking on. It's actually, I've been doing things through this graphical user interface, this GUI, but it's been doing things, it's showing you the commands that are actually going into doing that. And when you save your files, well, one thing, you can replay each step that it goes, and it'll go step by step through each of the different things that you like clicked and how things changed. And it'll also allow you to, if you do this, oh yeah, there's a bunch of alignments and things like this you can do. You can do a 3D print your structure, but what you do is if you just like, if you go to share link, you can see that you can actually copy a link with all of those commands. And so if you go and you paste that into another tab, well, it's going to go through all of those commands that you did down here. And it'll also give you these commands that you can go and copy and paste into Jupyter Notebook to do it in a Jupyter Notebook and have it display. What's really cool is that these are like, these links, so that these just, even without the URL with all those commands, you can get a link that'll take people to the image that you've generated. And you can embed these links into a website. So we're going to be embedding these structures, the interactive images, they're not just images, but like interactive images into our like course web page thing, so that we can all have this like gallery of images, which is going to be really cool. And yeah, things like that. You can then share this with like me so that I can see your structures. If you go to file, and if you want to like save a PNG, you can do that. But what's really cool is this isn't just your typical PNG, it's a special one. And so if you go and you save that PNG and then you open it back up in here, you can basically open a file, an iCn3D PNG image, and it's going to have all of that information embedded in it to get you back to where you were before, or to get someone else back to where you were before. And then you can also, when you do that, save an HTML, and then you can go and click on that HTML and get taken back to the session again. So really, really cool stuff, and a lot of different stuff that you couldn't do with like PyMol or ChimeraX. But there are things in PyMol and ChimeraX that you can do that you can't do with this. And so if you really want to go into structural biology, I recommend learning those tools. Or if you want to make really high quality figures, I recommend those. But for really just getting a sense about molecules and getting a better understanding, getting familiar with them, getting a quick look, iCn3D is a really good option. And so we're going to be using it extensively in my classes. And I want everyone to get really familiar, really comfortable with just every time you see a PDB code, go plug it in. It's really simple. You don't even need to download any software, and you can take a quick look. You can see all the annotations, all that good stuff. And so hope this helps you get comfortable with starting in. And I will use the links so that you can go through a couple of quick tutorials. I'd like to really thank Henry Jakubowski for helping teach me iCn3D and showing me all the cool things it could do, as well as for him and Patricia Flatt making this great resource available, The Fundamentals of Biochemistry. It's a free online textbook that I'm going to be using portions of throughout my courses. It has iCn3D. It has intro tutorials that I will link to, as well as it has embedded throughout all of its content. It has various iCn3D interactive molecules. So in some cases, you'll be able to twist and rotate them directly in the page, and other times it pops up into a new window. But then you're going to be able to play with it just like you would in iCn3D in the full screen thing. They also have this really awesome gallery. So this iCn3D molecular models gallery with lots and lots of different structures that you can then go and you can investigate further and play around with them yourself. So all of this is really, really awesome, and I will post links to all of it and hope that you find iCn3D cool to use too.